Hello, welcome to Biome 1005 Microbiology, and in this presentation I'll be covering the basics of taxonomy of microbes. So what are the learning objectives? To be able to understand basic taxonomy, nomenclature and classification of microbes, and to be able to describe various techniques for identifying bacteria in a medical microbiology context. So first of all, here are four definitions you're going to need to know. So taxonomy is the study of general principles of scientific classification and naming organisms, and classification is the organisation of organisms into progressively more inclusive groups by either phenotypic similarities or evolutionary relationships. Then you have nomenclature, which is the systematic naming of organisms based on international rules according to its characteristics. Then finally, identification, which refers to the practical use of a classification scheme. So first of all, what is taxonomy? So taxonomy is used in a microbiology lab in regard to diagnosis of infectious diseases. It helps to show relationships between different organisms, and it can be used to predict properties and behaviours of new species. So let's just give an example of the basic taxonomy of a human. Okay, so if you look down just this side, we've got life, domain, kingdom, phylum, class, order, family, genus, species. But you're going to need to remember this. Okay, so what I want you to do, I want you to try and make up your own mnemonic to try and remember this. So it could be something like Little Donkey Kong's Princess, blah 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 blah. So you get the idea. Just try and make like a funny mnemonic to help you remember these. So if we just go down this list and we'll try and figure out what the taxonomy of a human being is. So first of all is the domain name, eukarya, Animalia, Chordata, Mammalia, primates, hominidae, and this is the two most important pieces of information: Homo, sapiens. Okay, so the two names we give to an organism is the genus followed by the species. So it'll be Homo sapiens. Okay, so that's how we name organisms. Okay, and what that's called binomial naming. So we're using two words that are specific to that species. Okay, when it comes to binomial naming, there are just a few rules that you need to obey by. So the genus, so in this case it's Homo, always begins with a capital letter. Okay, and the species with a lowercase. When typing this on the computer, you need to make sure it's in italics, or if you're handwriting it, must be underlined. It just makes it easier for the names to be identified. Okay, when it comes to classifying bacteria, we can do it in a number of ways. We can either do a phenotypic analysis, which is regards to their morphology, metabolic, physical and chemical characteristics. We can do a genotypic analysis, which is the characteristics of their genome. Or we can do a phylogenetic classification, which we can use to establish evolutionary relationships between organisms based on a small subunit of RNA which is located within them. Okay, so these are the different ways we can use phenotypic analysis to help identify bacteria. So we can test their morphology, or whether they get gram stained, the motility, so however they move, metabolism, physiology, cell chemistry, and other traits. So for example, antibiotic resistance, which are specific to those microorganisms. Okay, so this diagram here is just showing you just a few examples of colonial morphology. So by that I mean, if you look at an agar plate, what you can see on the agar plate. So if you're looking down at it from a bird's eye view, you can get circular colonies, irregular, filamentous, or rhizoid shaped colonies. So quite a few different shapes there. Then if you look at it from the side view, is the colony raised? Is it convex shaped, flat, imbornate, crater formed, or even concave, so like reverse of that? Then if you look at the margins of the colony, so the outside areas, is it entire, so it's like a nice smooth undulate where it's got these small lobes, filiform where you've got like filaments springing out, curled where you've got waves coming off the end of it, or if it's lobate in which you've got large lobes. Okay, so you can literally get any combinations of these colonies. So for example, you could get a circular, flat, lobate colony, for example, and that's specific, and that could be specific to that bacterium. Okay, now this is just going to show you the individual morphology of bacteria. So for the cocci. So you've got cocci, or just coccus, which is an individual cocci. Diplococci, 2 streptococci, which is when they line up like that. And then you've got like tetrads, staphylococci, all sorts of examples. Same for the bacilli. So a coccobacillus, which is halfway between a cocci and a bacilli. A normal bacillus, which is an individual one. Diplobacilli, etc. So you've got all sorts of different morphologies of bacteria. And also we've got spirochetes here, which are really unique, and we'll be covering those in a later presentation. So here's just a few methods that we can use for identification in regards to their phenotypics. So this here is called the agglutination test, which is basically we've got a sample of the bacterium and we've got antibodies which are corresponding to those. If they agglutinate to them, what you get is that you get these clumps forming around them, and that's when these antibodies attach to the bacterium. You can also do serological typing, in which you can find specific antibodies for a bacterium. So for example, we've got E. coli here, in which we'll have 0157H7, which is specific to that bacterium. We can also do immunofluorescence, so this direct immunofluorescence in which we stick on an antibody which has been tagged with a fluorophore. Or we can do it indirect, when we attach an antibody, the primary antibody, and then to that we then attach other antibodies which contain the fluorophore. Okay, we can also do tests for viruses. Okay, so what we can do for a virus is something called an ELISA, which stands for enzyme-linked immunosorbent assay. Okay, here on the left we've got a direct ELISA. So here we've got viruses which we have placed on a surface just to keep them still. What we can then do an influx of antibodies, and these antibodies will have enzymes attached to the base of them. Depending on what antibody fuses to this virus will give us a specific colour change due to a substrate and enzyme interaction. Okay? And depending on what colour change we get will therefore tell us what virus this is. We can also do an indirect ELISA, in which we get the virus, we then attach the antibodies to them, and then onto these antibodies we then attach a secondary antibody. And this secondary antibody is what's going to contain the enzyme. And again, depending on which antibody fuses, will give us a different colour change depending on that. For bacteria, we could also do DNA sequencing. Okay, and what this will do, this will then produce bands of DNA. If we compare this to our unknown bacteria, we can therefore match them up. If there is a complete match, we can therefore deduce that that specific bacteria is what's causing the disease. For completely unknown bacteria, what we could do is something called nucleic acid hybridization. Okay, so again, we use DNA sequencing to do this. If 70% or more of the DNA is similar between the two, we can deduce that that is of the same species of what we've got. If there's between 70% and 25% similarities, they're in the same genera, but they're in different species. If less than 25% of the DNA is shared, then that means they are of completely different genera. So I bet after all this, you're probably wondering, what is the importance of taxonomy in relationship to disease diagnosis? Well, the answer to that is that we have two categories of disease agents. We've got specific and non-specific. Specific disease agents are different microorganisms that cause a specific disease. Okay? So for example, we've got Y. pestis, Yersinia pestis, okay, which is the causative agent of plague. No other bacteria can cause that, so as soon as you see the symptoms of plague, you can be like, right, that's Yersinia pestis, okay, let's treat it quickly. Then on the other hand, you get non-specific. Okay, this is when you get different microorganisms that cause the same disease. Okay, so S. pneumonidae, H. influenzae, Staphylococcus aureus, they all cause pneumonia. Okay, so just based on symptoms alone, we cannot differentiate between all these. We'll have to do further tests to differentiate between them. Okay, and now finally we've come to the test yourself section. Okay, so in this section, I'm not going to give you the mark scheme. I'm going to tell you how many marks it's worth, and I want you to write your own mark scheme for these questions and answer these. Okay, so first of all, define taxonomy for 2 marks. Then for 8 marks, Name the different sections of the hierarchy of taxonomical divisions and make sure they're in order. Then finally, an essay-style question. Give examples of different bacterial morphology, including colonial morphology, and explain how these can be used to identify bacteria. And that's worth 25 marks. Okay, so I hope you've enjoyed this video, and make sure you guys leave a comment, and if you want to, leave a comment for questions you want other people to answer. So, thank you for watching, and good luck revising. Peace.